For everyone with an interest in NASH, or more broadly, fatty liver disease, Surf's Up, Season 2, Episode 36 of Surfing the NASH Tsunami. Our wrap-up for ILC 2021 starts now.
Today on Surfing the NASH Tsunami.
Ian commented the other night, we need to make this as simple as possible so people can follow pathways to try and identify the patients. And EASL has greatly helped with the new non-invasive guidelines, which can filter out the right referrals and the more complex ones to hepatologists.
But then when you're thinking about the drug development pipeline, we also saw some really interesting things. The Maestro-NAFLD-1 study, a great example of how the field should be, requiring patients with a non-invasive test. You're following them through on a therapy and you're assessing efficacy on non-invasives.
Right now we randomize based on type 2 diabetes, and the question is we should randomize based on these genetics variants and look at the placebo response based on that.
Last week, close to 6,500 stakeholders from across the global hepatology community convened virtually for the Digital International Liver Congress 2021. Today, join hepatology researchers and key opinion leaders, Dr. Stephen Harrison, Professor Quentin Anstee, Dr. Mazen Noureddin, and Professor Ian Rowe, plus liver wellness advocate Louise Campbell, and pricing and forecasting guru Roger Green as they explore some key topics from last week's Congress. Today, on the Surfing the NASH Tsunami podcast.
So we have Stephen back from vacation. Hey, Stephen, how are you today?
Hey, I'm good. Glad to be back.
And Stephen is going to show you, we're going to talk about Stephen's homage to surfing on the beach in Panama City in a couple of minutes. And we have Quentin Anstee with us for the first time in over a year. Quentin, I'm delighted to have you here again.
Thanks very much, Roger. Great to be here.
And we've learned during the introduction that Quentin and Louise have a fascinating and occasionally sordid history, some of which they will tell you about in just a moment. And then we have Mazen Noureddin and Ian Rowe, who've met each other for the first time today over video. Hi, guys.
Hello. Hi, everybody. Pleased to be back.
And then we have me. If you could see me, you'd know that we tried to get our studio in Texas finished for this podcast. We didn't do it. So I'm sitting in a room with a blanket draped over my microphone so that I can have the fan on because it's hot here. And wearing the same clothing I was wearing all day when I was trying to put this thing together. You can't see that, only my baseball cap. And there's no video today, so that's a good thing. So before we get started, I want to do our icebreaker in a slightly different way, which is Stephen has a story to tell, and Quentin and Louise have a story to tell, and then Mazen and Ian and I can come up with whatever we come up with that qualifies as a story to tell from whenever. Quentin, why don't you and Louise go first?
Okay, well, so it's a little known fact that for many years ago, Louise and I both worked together in London at St. Mary's Hospital, and I'm claiming responsibility for getting Louise hooked on FibroScan back then, where in order to make the business case to allow the charity to buy a FibroScan machine with an XL probe for us, we went into the boardroom armed only with a Mangini needle and a large citrus fruit and biopsied it on the table to demonstrate the need for non-invasive tests. Louise, over to you.
I think that we did look for a watermelon because we were looking for something liver-sized, but we came up, the only thing in the St. Mary's High Street of Trade Street was a big grapefruit. And actually it was probably better because as you tear the skin of the grapefruit, it's not dissimilar, sadly, to a liver biopsy. And there must have been 25 people around there making this decision. And as soon as they heard the crunch of the needle go through, we knew we had £100,000 from the charity. And that was back in 2007, I think, Quentin. So it was one of the first FibroScan machines into the country, and the charity at St. Mary's are massively important and basically funded the whole FibroScan service for many, many years, and all of the patients got great care because of that. And yes, he is accountable for why I now do a lot more with FibroScan, taking it out. So yes, Quentin and I are responsible for the grapefruit incident, I suppose.
So we're going to go from a grapefruit in London to Corona Lights with lime and artistry on the Redneck Riviera in Panama City, Florida. Stephen, why don't you tell us what your personal best for the week is?
Well, look, what I've realized is that what I do artistically is directly proportional to the amount of stress I have in my life. And normally about once a year, I'll go to the beach, and I used to do sand castles. Now I do something that I just call sand art. It's whatever comes to me, and I'll make it on the beach. beach. And usually I only do one. But again, noting the stress in my life, I actually did this 3 days in a row. I don't know what I'm going to make when I do it. I just pile up a bunch of sand and then start sculpting. So my favorite one this year was Herbie. If you remember the little beetle, the movie with Herbie, it's a little VW bug. Number 52 actually was on the side of it. And that's what I attempted to make this year. And I shoved a surfing board down in the convertible and off we went. So we don't have video to show you, but it was certainly a stress relief for me. It took about 6 hours to do and a lot of fun. So the other 2 items I made was a couch to sit in that was pretty big, and then a wave with 3 dolphins coming out of it. At the end of that, my hands were blistered and my knees were raw from, moving around the sand on my knees, and I was done. But it was fun. It was a great time. Share the secret, I did that between a couple different lectures for Easel, by the way. In between. Nobody commented that I wore the same tie and the same shirt for 4 days in a row, but, it was okay. It was good.
So, Stephen, that's great. And in fact, with Stephen's permission, we will be posting pictures of Herbie, although there is no number 52 on it that I can see, and the other 2 items on the website by the time this episode goes live on Wednesday night. The athletic accomplishment of the week, I think, goes to Ian, since I've read about it already on Twitter.
It's not my accomplishment, actually. It was in contrast to Stephen's stress relief. I spent yesterday morning in a state of very high stress as my oldest boy played in the regional cricket cup final and his team came out winners, and it's a sort of culmination of an already quite long season and a sort of personal journey for him as being the youngest in the team and, contributing to the overall success. So the next match will be in the north of England, semi-final, I think, against Northumberland, which is near Quentin, and where we'll be on holiday in 2 weeks' time, where there are very large beaches, but I suspect my contribution will be much smaller sand sculptures than Stephen's managed.
I've been there. I know the River Tyne there. There's a beach right there, Quentin, right?
Absolutely, yeah. I remember you, Stephen, obviously not in shorts on that occasion because the hail was coming horizontally.
That's true. But, hey, look, there's sand. You can do something. See what you can do, Ian. Let's throw it up there on Twitter or text me or something. Quentin can make a speedboat, too, so we've got to see what that looks like.
Actually, I would like to invite any of our listeners who think you can outdo the work you're about to see to please send us what you've got. And I'll come back to the idea of live audiences in a minute. But if you come up with a sufficiently compelling sculpture and you want to come on live to tell us about it, we'll find a way to do that. You'll see more on that later. Okay, so Mazen, the only people who don't have something really interesting going on that I know of are you and me. So what you got?
My 8 weeks old daughter got her vaccine on Friday, and this is the 8 weeks, so they get a lot. And the best news in the last 2 weeks that we're enjoying SNOO. If you know what SNOO, SNOO is a bed that shakes electronically, and if the baby cries, it shakes harder and put them to sleep. So that has been very helpful. And it also makes white noise, which is kind of hair blow dryer that calms them down. So this is my life right now. But this is my exciting news. So, my wife is Belgian, and my mother-in-law grew up in Belgium, but she's Spanish, and they actually own a vacation house in Altea, Spain, and that's where I'm going August 6th. So I look forward to that.
Mazen, if I catch you in that SNOO, we're going to take a picture and post it.
My hand would not fit in that SNOO, Stephen.
So on days when your hands are jittery, put a hand in the SNOO and let us watch it calm down. How's that?
I'm going to send you a picture of that SNOO, and my daughter is shaking and has been very helpful, that SNOO thing. Google it. It's really neat.
I have to tell you, I think this gallery of pictures idea might become a permanent fixture of the podcast, or at least whenever anybody has anything interesting enough to post a picture on. I actually have spent the last day trying to put together last 6 hours trying to put together my Texas recording studio in Corpus Christi. Hoped I would have it done in time for this podcast and that I could, by video, show at least my fellow panelists what a cool thing, because I don't normally do that kind of stuff. I'm just not terribly handy at that kind of thing. But this is going along fairly well. It's just taking a lot longer than I thought. So next time I'm in Texas, I'll be able to show you the studio. For today, all I can do is tell you, and if you can't see this because we're not on video, but I'm using a blanket on top of my microphone in an attempt to muffle the fan that's keeping this room below 85 degrees of circulating air. So not exactly what I had in mind, but it's what we're going to go with. So with all that, I think one of the more interesting icebreakers we've ever done. Let's dive into the podcast. The purpose of the podcast is to kind of wrap up and say farewell to ILC 2021. We spent a bunch of time over the last several days, a total of about 4.5 hours in 3 different sessions, Thursday, Friday, and Saturday, talking about and hashing over various articles and things of interest. Louise was there for one, Ian for one, Mazen for one, Stephen was doing sculpture, and Quentin was too tied up in the meeting, but was good enough to join us us this evening. Basically, I listened to all of our tape again yesterday while I was flying from New York to Texas and came to the conclusion that there were really 4 sets of issues. At least you could break it into 4. You could also break it into 7, but I don't know how to do 7 issues in an hour. But there was a lot of discussion about the general issue of diagnostics, both non-invasive and also some things about the AI and machine learning. There were obviously presentations on drugs and development. There were presentations on patient populations, some on epidemiology, some on genetics, and then broadening the focus of treatment, a lot on multidisciplinary care, and a couple of really interesting general session papers, one on food insecurity in the U.S. and one on the value of alcohol regulation in morbidity and mortality in Central and South America, I think each of which came up with at least one major theme. On this podcast, we talked about a few of the, on the first one, we talked about a few of the litmus presentations, and then in terms of drug development, we, on Friday afternoon, talked about a whole bunch of diabetes drugs in metabolic contexts, semaglutide, lanifibranor, and then Ian, continuing his ability to find the really interesting and unique needle in the haystack, talked about a canonical Flozin poster and SGLT2s, which I thought was great. And then on Saturday, we talked about Stevens, efruxifermin, late breaker, and one other that I'm not remembering right now. In terms of the epidemiology and genetics, I talked about Laura Safadi's paper on lean NASH on Friday, and someone on Friday talked about another one of the papers about rare events in NAFL populations. And then Louise talked about the multidisciplinary care. We all talked in different ways about the food insecurity paper. And so we touched on all 4 of those in the last 3 days. I'd like to see if we can talk now as a group about what we think were the most interesting things and the key takeaways on each. So I'm going to throw a topic out, and then I'm just going to ask the group. Let's start with the whole issue of diagnostics, non-invasive testing, AI, the various litmus presentations. What should our listeners take from that body of presentations? Where do you want to go first?
I think it was positive. I think there were several different sessions on non-invasive technologies being put in primary care and various pathways. There was a couple last year, but I think Mead Matthews' primary care pathway down in Southampton was good. John Dillon followed her about making it simple. And Ian commented the other night about we need to make this as simple as possible so people can follow pathways to try and identify the patients. And I think EASL has greatly helped with the new non-invasive guidelines, which can place, if primary care in the UK, for example, get access to it, FibroScan straight at the heart to filter out the right referrals and the more complex ones to hepatologists and keep the others in primary care. The only reservation I have that is that would be great if we actually had an appropriate system in primary care to support weight management, lifestyle, fitness and health, which we do add in a little bit more when we go into a hepatology or endocrine setting. But we know it doesn't work too well in primary care, so I would like to see primary care funded better to be able to offer those better options. Certainly for me, non-invasive technologies are moving forward. I think there was a lot of evidence from Litmus on how strong FIB4 is. Again, the EASL guidelines supported that. We're moving in the right direction. We are moving away from biopsy, but can we move completely? No, but I think it will be a combination of lots of non-invasive technologies, and there were some interesting biomarkers discussed by other people.
So Quentin, go ahead.
Yes, I'd agree with what Louise said. If you pull pieces from several of the abstracts we heard presented during the course of the meeting, there are a number of different things there that give us a clue to the direction the field is moving. So if you think first of all about diagnostics and identifying the right patients, some of the data coming out of Litmus, so comparing a range of wet biomarkers was very good. Similarly, the data that Stephen and Michael Pavlidis in Oxford generated looking at the use of FIB4 followed by FibroScan, all very good for that diagnostic context of use. But then when you're thinking about the drug development pipeline, we also saw some really interesting things. So if you think about the Maestro NAFLD-1 study, a great example of leading the way to how the field should be, the idea that you're acquiring patients with a non-invasive test, you're following them through on a therapy, and you're assessing efficacy on non-invasives. Now, we can debate whether it was perfect, where there were issues, and so on and so forth. But I think that was a really nice paradigm demonstrating that and shows the way to how we can use things in practice. And then the third strand that I took away from the meeting was about endpoints. So we had Vincent Wong presenting the data on the FAST score on semaglutide and its ability to detect change, which isn't something we've seen before. It was designed as a diagnostic, so that's important. And then also some of the other semaglutide data looking at the intersection of different biomarkers where you're using ELF score, FibroScan, and histology and looking to see the wisdom of the crowd. Are they all going in the same direction together? So I think there were 3 strands there, but you have to dig deep for them.
I'll just build on that. Quentin, you did a great job of summarizing the context of use of these biomarkers. And just adding to that, just starting at the endpoints piece where the FAST score was brought in, I think Rohit showed data from the combo cilofexor forsocostat data looking at the similar set of data. So showing that drops in FAST actually correlated to improvement in fibrosis. So that was nice to see a kind of a completely different drug, completely different MOA, completely different patient population, using the same tool, showing similar type of data. Efruxifermin had 2 posters, in addition to the late breaker, that looked at that specific context of use as well, but combining MR-PDFF with ALT and showing that, yes, ALT, 17 units per liter reduction did correlate with improvement in NASH resolution. However, not fibrosis benefit. And there's some nuances to the trial design, I think, that explain that. But when you put PDFF reduction alongside ALT of 17 units, you actually enhance the ability of those 2 tests to predict NASH resolution, fibrosis improvement. In fact, we've talked about PDFF reduction of 30%, then we've gone to 50, then we've gone to 70, and now we have liver fat normalization. It doesn't surprise me that if you achieve liver fat normalization and a 17 unit per liter drop in ALT, that's the holy grail, right? That's where we all want to be. And this trial, although small, its inherent limitations considered, suggested that. So I do think the crystal ball is beginning to show us how these tests could be used. And quite frankly, just following on the emerging topic conference we had with AASLD the week before, where you have Frank and Annie sitting there and encouraging people to look at these non-invasive tests, not as a surrogate of a surrogate, but as a surrogate. Link it to outcomes, show us some benefit, and then we can move away from biopsy. In fact, provocatively, I asked, what about an ELF score greater than 11.3 at baseline? Or what about using MRI-PDFF as a marker for outcome? Because it kind of spans the waterfront. If your mechanism of action moves liver fat, you can use it to diagnose, potentially. You can use it as a marker of therapeutic efficacy. And to Quentin's point, all we need are 1,000 patients worth of MRI follow-up data to show us outcomes. And that's coming. So fantastic way to summarize it between Louise and Quentin, but just a couple extra points I wanted to throw in.
That's great. Mazen, Ian, anything to add?
I'll just repeat, in addition to the data that everyone talked about, I also like the Agile 3 and Agile 4 data that I think added strengths to the FibroScan. We all had issues previously with stiffness of 14 and stiffness of 10. We wondered if the stiffness of 14 really represents cirrhosis and the stiffness of 10 represented F3 or whatever cutoff you want to talk about. So that was presented earlier by Jérôme Bossier, Darwin Sanyal, to strengthen the rule-in and decrease the indeterminate zone using a combination of the stiffness and clinical parameters. And Vlada brought a very good point that the area under the curve was not quite optimized, was slightly higher, but yet the indeterminate zone was decreased using these 2 scores. And I brought up the point that the positive predictive value was very good, but these cohorts were a little bit biased in terms of the prevalence of the F3 and F4, which is a point that everyone should pay attention with when they talk about PPV. But I thought it's a step forward in terms of improving imaging data and combining them with wet biomarkers. Those were very good 2 abstracts.
And I think, Mazen, what you've just said really highlights the difficulty in the short term, and that is tying the non-invasive test to the biopsy. And that conversation that was had at the Emerging Topic Conference with Frank and Anya, and then the points that Patrick Bossett made in the Litmus liver screen session on Thursday morning, talking about skipping over biopsy as the reference standard. The sooner we can tie the non-invasive tests to outcome, the better. because we'll no longer have to worry about thresholds, particularly because they'll give us a probability of events and hopefully what the impact of therapy will be on those probability of events. And I think the sooner we're away from biopsy, the better, because it'll simplify a lot, simplify management in practice, and it'll also simplify trial design.
So I agree with that. And one of the things I think that's really encouraging is to think about going straight from non-invasives to outcomes instead of having to match non-invasives to biopsy. I promise I won't talk about this anymore after today because I've talked about it each of the last 3 days. years. But I'm mindful of the drug Premarin, which back in the early 90s was 55 years old, selling $2 billion a year, and no one could come up with a generic for it because it's mare's urine. And the pharmacokinetics of mare's urine developed in 1939 were so poor that you couldn't replicate them. The error margins were too big to match. So therefore, there was never a generic Premarin. I mention that because I think the same is roughly true of biopsy for a variety of reasons, some of which are idiosyncratic. The error margins will eat you alive. So if we can go straight from non-invasive to outcomes, then in fact, we're talking about going from the test we're using to the thing that really matters. If we have to stop at biopsy on the way through, I think that's highly problematic. And Quentin, maybe I think I read this right. One of the things I took out of this meeting was more and more papers on not having to do that, just going straight from test to end instead of having to stop in the middle.
Absolutely. I think it is definitely the way we need to move. I think Litmus is generating data. You're going to see a lot of data coming out of some of the big phase 3 studies that are going to help answering this. It is the direction the field needs to move because, as you say, histology, there's sampling error, there's observer variation, as Stephen demonstrated very nicely in the Davidson paper. It's troublesome for the field. It also means when we're tying to biopsy, I don't think we're ever going to get an error meaningfully beyond about 0.85 with a moving goalpost.
The question is, I guess now we have enough data showing correlation between NIT's progression and worsening outcomes. The question is how much more the regulators want from us to show, is it really improving NITs lead to improving outcomes? Is that the only piece that they're waiting on us, and how long that's going to take? I don't know what is missing now to start implementing NITs as a primary endpoint. For instance, why not implementing PDFF plus improvement in stiffness on fibroscans plus ELF as a primary endpoint for phase 3 study now? And a lot of people will argue it's too early or not, but why not? Why not start the conversation when is this ready as a composite endpoint? So
I think part of the issue, Mazen, Frank alluded to, it's not in their wheelhouse. It's not in the hepatology division of the FDA to look at non-invasive. It's almost like he was mentioning that there was stovepipes. There's the whole biomarker qualification program run by the CDIR, I guess. It's a completely different branch of the FDA that Frank is in. So what we need is maybe what he mentioned, is bring that other division to the table with Frank and Joe Turner's division, and let's have a conversation about how we can work together to generate the answers we need to generate to move beyond liver biopsy. As you mentioned, we already have outcomes data. I mean, you can begin to connect the dots. We've done that already. We've shown that if you improve fibrosis, you improve outcomes. Arun presented that data at AASLD. We also have data from Alina Allen and yourself that MRE is linked to an outcome. We have that data with CT1. Multiparametric is linked to an outcome. ELF is linked to an outcome. But what we're missing here is that little dot connecting fibrosis regression with a magnitude of effect change on MRE, CT1, and ELF score that then connects the dot to outcomes. But that's super easy to generate.
Correct. But my counter argument, I'll ask them, then why you have NASH resolution and histology as an acceptable point for the subpart H approval and not letting others that they are NITs that they are now correlating with outcomes be a point for approval. Anyways, I just open can of worms that we keep going back and forth in emerging topic conference and all that. So it's a topic of debate that's worth keep talking about.
Well, first of all, the more we can have these conversations, the quicker that resolution is going to come. Whether it's on this podcast, whether it's at the single topic conference, whether it's at EASL or AASLD, or side chats with Liver Forum, or side chats with the FDA when we all go to the FDA for certain pharmaceutical companies, those conversations need to be had. The drumbeat will get louder, and eventually there will be a pendulum swing, just like there is with standard of care, Mazen, right? I mean, you want to know what makes anything standard of care. It's not one publication in a guidance document. It's not one New England Journal paper. It's when that rising tide lifts all boats, when that news becomes so ubiquitous that one day you turn around and it's like, how did we get here? I don't know, but that's what we're all doing.
Yeah, so you see my point, the NASH resolution, why it's a point of approval. Why not 2-point NAS improvement would get you approval?
The issue here is that at the point that the current histological endpoints were framed, which is now 5+ years ago, they were the best that were available. So they were a reasonable, pragmatic choice. However, in the intervening period, there has been exponential progress in biomarkers. And so just like you revisit the guidelines, maybe we're approaching the point where it's time to open these conversations again and revisit them.
Totally agree.
And then you go back to where Stephen was a couple of minutes ago, which is that for us to have that conversation is easy, right? We have venues to have it in. For FDA, it's a lot harder. For regulatory agencies, it's a lot harder. You have more inertia to deal with and more different ways people spend their times. And then if you put the biomarker tests and the drugs in two different places, then there has to be enough energy to make that dialogue actually push forward. So I agree with Stephen said. The best thing we can all do is have the conversation over and over and over again with them, with ourselves in places that folks might be listening to. Go ahead, Quentin.
Completely agree. One of the things where the field possibly does itself a little bit of a disservice is we now have such a wealth of biomarkers that we haven't yet coalesced down to a small number of biomarkers and said, well, these are the standard we're going to follow. And the moment we started to coalesce around a few, that would be very helpful. Even looking at those Maestro-NAFLD studies, there were actually subtle differences in the biomarkers presented at the end point between the serotic and the non-serotic arms of the same cohort. And that was one of the things that litmus presentation was doing, where it was comparing so many to FIB4 as a standard. It was really to provide a yardstick whereby we can say, well, these are all pretty interchangeable, so let's select some of these and go forward with them. And, we're going to do the same with imaging. We'll do the same with various other things as we go forward. But that's part of that dialogue.
I think that's a very important point that Quentin is making. We have to make it clear to everyone that the field has moved. Networks like Litmus has done a great job in validating biomarkers, and there's certain ones that has shown that it can do the job longitudinally, and there are certain ones, ELF, MRA, FibroScan, that have shown correlation with outcomes. Not everyone, but there are certain ones that we can rely on. And I think by combining few, we can increase our confidence that we can rely on this composite endpoints to reach where we want to be.
All right. So first of all, I think that's a great place to close this portion of the conversation, because we've got 3 more topics to get to and limited amounts of time. So next, drugs and development. Overall consensus, how promising / exciting did you find what you heard in this meeting? How far do you feel we moved the ball up the field? And what do you think we've learned, either about individual compounds or about ways to think about using drugs to treat the overall disease? Let's start with the last one. How far we moved the ball upfield, and what does that look like?
I can give it a shot before I move it to others. What was presented in this meeting was a reflection of the COVID-19 year. It's not like the previous meetings that we saw multiple presentation of new drugs. There was the cirrhotic trial that Stephen presented, which was new, and 1 or 2 new others. It was mostly a secondary presentation from Lani and semaglutide and cardiovascular or the fast core longitudinal follow-up, but not as many as the previous meetings. And that's expected after a slower enrollment and some static enrollments and trials. But I thought it was still very good for a year after COVID. So overall, I think we'll see more in the next meetings and more outputs. The NGM, probably they'll present their data in the coming meetings. BMS we'll see more. Stephen is more involved in those, so maybe he has more input.
I agree. I mean, it was kind of an uncharacteristically short yardstick for prospective data, certainly less than what we've been used to seeing at prior congresses and so forth. And I totally agree that it's probably due to COVID. I mean, there's no doubt that enrollment shifted to the right at least 4, maybe 6 months for these trials. I mean, Quentin, we see that even with Litmus trying to get these imaging studies done. So, I mean, it is what it is. I would say that we're primed for some big news at the next Congress. One way or another, there probably will be multiple different mechanisms of action being presented from the clinical side, and probably more data on some combination work as well. Where I would say we move the goalpost, and maybe this is me being a little biased, but I do think for the first time ever in NASH, outside of bariatric surgery, we showed that we can take a well-compensated cirrhotic patient and we can improve their liver disease by at least 1 stage in a third of the patients. And I absolutely know and expect there to be naysayers to that, and that's absolutely fine. That's the way science is, and that's the way we get better. But just in the defense of that, every non-invasive test that we've talked about today got better. And to that point, we now have a magnitude of effect change that we need to reach at least a preliminary bar that we need to reach to show a regression in a cirrhotic patient. Maybe it's 9 for a Pro-C3, or maybe it's almost a full point for MR elastography. I don't know, but that data is beginning to come out. It's early days, small numbers of patients, but I think there was some very helpful data, encouraging data that we could move the needle in a well-comp F4 patient. That was exciting. Where we go from here, I think what you're going to see is you're going to see more mechanisms being studied in cirrhotic populations. We have the reverse trial. We have the BMS Falcon 2 trial. We have the NGM F4 study, Alpine 4. And now we have an early small trial with semaglutide. But beyond that, there hasn't been a lot of interest in studying the NASH population. And I left out galectin with belapectin also moving into adaptive phase 2b3, although their endpoint is more of prevention of the development of varices rather than regression of fibrosis. But where do we go with the cirrhotic cohort is going to be very telling. You'll see a lot of movement coming out of this meeting as we move into future meetings with companies really trying to get after a cirrhotic population, because in my mind, that might really jumpstart the non-invasive test. It's easier to link an F4 non-invasive test to an outcome than it is an early stage disease. So maybe we prove it there. We get a drug approved on a non-biopsy endpoint in a cirrhotic, and then we reverse engineer this thing back to a non-cirrhotic population. But we could sit here and pontificate what's going to happen with the data, but I think it certainly brings up a lot of positive conversations relative to cirrhotics.
So first, let me tell the audience that when Stephen made the point about research with cirrhotics speeding the path to non-invasive testing, I saw 2 head nods and a thumbs up out of 5 people, which isn't unanimous, but was pretty good support for the point. Whoever else, I have a question, but if anyone else wants to go on, I will step back before I ask it.
I'd just add one thing. I agree completely with what Stephen's just said there. The other study which was interesting was we're starting to see data, for example, targeting HSD17B13, and that's appealing because we're now taking genetic signals that we've identified and we're targeting those. And I think that's going to be the next wave of treatments. So again, tiny proof of principle studies, but hopefully we'll move that forward.
And on that vein, Quentin, just to follow up, that was in 5 patients, I believe. Was it more than that?
If that, it was mainly preclinical and then a very small number for safety.
ALT reductions were something to talk about.
Yeah, and that's the point. And I suspect for mechanisms of action like that, we're going to have to be much more focused on who we recruit into studies. Maybe it's going to be gene-driven. We know, for example, HSD17B13 interacts with PNPLA3. So it may be that there are subtypes there we need to chisel out for these sort of treatments. They're probably not going to be as tractable on a large scale as many of the others, but it's very exciting to see those beginning to come through. And of course, there are others which weren't presented at this meeting targeting similar genetic points.
Yeah, you just struck a chord with me and made me think about something. In all of our non-responders to drug therapy, whether it's a non-responder to an FGF21 or an FXR or an FGF19 or the THR beta or whatever, we've never done genetic testing to link the responders versus non-responders. And I suspect there's a story to be told there, and potentially linking that to future studies where, if they're enriched for those particular polygenic risk scores, we study them using RNAi or oligonucleotide therapy or whatever.
Absolutely. I think that's the way the field needs to move here. And particularly where we have failed trials, to have the courage to do a deep dive, or not even the failed trials, just the trials that didn't quite deliver what we'd hoped that they would deliver, and start looking for subgroups, but in a very scientific and objective way.
I totally echo that, not to mention the placebo response. Right now, we randomize based on type 2 diabetes, and the question is we should randomize based on these genetics variants and look at the placebo response based on that.
So first of all that makes tremendous sense to me. Second, I have a completely different kind of question, which is we've talked a lot over the last few months about both the regulatory availability or acceptance and the clinical value for F2 and F3 patients finding drugs that might resolve NASH without having an impact on fibrosis. If you simply stop fibrosis for a cirrhotic patient, How much benefit do you believe that will provide? Is that a realistic expectation for some of these drugs, that maybe they won't have an impact or have a fast impact, but they might actually stop the process? And how do you think that will be looked at regulatorily, in addition to how it would be looked at by the physician community and the leader community?
The question that you ask raises a whole load of questions in light of some of the conversation we've just had about the identification of non-responders. I'm not certain that I know what a non-responder is against a histological endpoint where there's a lot of variability. And the benefits to shifting population risk of disease progression, where there may be some effective treatment, might actually be quite large, but we don't know that yet because we don't have the outcome studies to be able to tell. If you can stop progression of fibrosis and cirrhosis before patients have developed significant portal hypertension, then it's likely that you'll see a reduction in clinical endpoints in that patient group. And so in a clinical endpoint-driven study, you would probably see that. The impact on cancer risk, well, I think that's uncertain, and there are a lot of open questions about that. So those sorts of drugs probably will show benefit. Show as much benefit as a drug that's very active in reversing fibrosis, probably not, but they might still show a degree of efficacy in a longer-term study.
You know, Ian, if we're thoughtful about this and we surmise one theorem, and that is, it doesn't matter what insult you give the liver, it's going to behave very similarly in how it reacts to the insult. If you agree with that theorem, and then you have an alcoholic that presents to the ER with decompensating disease, and he has the come to Jesus moment where he quits alcohol. 6 months later, we know that about half of those patients will show significant improvement. Many of them no longer require diuretics, no longer need paracentesis. Their energy level is back. Yes, they're still sarcopenic as heck, but they feel better and their quality of life is improved. And quite frankly, we've all seen them go on to lead relatively normal lives. And certainly, had they not quit drinking, they would have succumbed to their disease. So that's a situation where you didn't regress cirrhosis. You just prevented it from killing the patient. And that speaks to the fact that if we can halt progression of disease through whatever mechanism, that'll have a positive impact on outcomes.
I agree with everything you guys are saying. If we take it to the patient population, we've heard Donna and all of the other advocates on here say that actually when we look at the FDA's ruling, that holding the disease where it is should be an outcome, because patients are comfortable with that. If we do not progress, there is more time and we can get the data that you're talking about. We can see if it leads to more endpoints. We can see if it regresses those endpoints. We can see what the cancer risk becomes. But if we're always trying to reverse it, we are missing the elephant in the room, which is actually just hold it, help the patient stay well. And we will see in most cases exactly what Stephen's described there and Ian, is that we will get less health expenditure. A lot of people will reach that moment of, actually, I do feel better. I am regressing. And each small step that's a positive is often followed by the next step. Yes, there might be some regression in behaviour, but I'm actually more for forward movement, not necessarily that you can't make a mistake. And I think that's where we've got to get to. We have to say that it's not all about cure at the moment. It's got to be about plateauing the disease, I suppose, and not progression. And Manal says it all of the time, that it really should be an outcome.
Thoughts, comments, or should we go on to whatever's next? Okay, good. Then let's stop beating the dead animal further into submission and go on to the next subject. So a third really interesting focus that we commented on a lot was that this meeting was a lot more multidisciplinary. It's been sessions talking about primary and parts of sessions talking about primary care, a lot more focus on other related medical specialties. The people who came on the podcast agreed that that was really valuable and important. I guess my question is, what do you see coming out of that going forward, and how does that convert into a change in practice or better outcomes of treatment for patients?
We need to increase awareness in our partners in endocrinology and primary care, because those where the patients come from. And this is the first step. It's partnering with them. And I think by implementing such a model, having multidisciplinary team is the right step. So I thought that was very well done to increase awareness.
Louise, you went to some of those sessions. In fact, spoke about some of them on the day you were on podcast. Anything you want to comment on?
I think I covered it in those sessions, and is that we are putting in different, and Quentin will know way more than I do about the local pathways in different areas of the country. There's obviously the Camden and Islington pathway. There's various pathways they're using, Nottingham, Southampton. At the moment, we're doing pathways, but there seems to be no joined up single pathway. Is that right, Quentin, or is it just John Dillon had another one?
You're right. What we're seeing is right now, care delivery is very patchy. If you're lucky enough to live in a postcode in the UK or a zip code in America where Stephen lives, you're going to do better than if you're somewhere down the road where maybe there's a hepatologist who's less interested or so on in fatty liver disease. And that's something we need to try to get away from. I mean, there's been a very nice opinion piece published today, actually, in Nature Reviews Gastroenterology, specifically looking at some of the care models that have been developed. His work that Jeff Lazarus has led. Again, looking at some of the different models that have been used around and potentially which of those may be more tractable. We're all trying to struggle with the same challenge. Different people have found different solutions, whether it's reflex testing for biochemical abnormalities that are done in, for example, Scotland in some areas, whether it's a different technique. And we need to try to learn and harmonise on those.
The other thing that did interest me was some of the population studies. There was a very good one out of Spain that really showed that, and they used FibroScan. I think it was a population of 12,000 from 16 GP practices versus 500 biopsy proven, and what they came up with was over 8% of the population had a FibroScan of over 8. So 5.6% of the population had a FibroScan of greater than 8, which is a bit concerning. When they drilled it down, 1.3% of the population had NASH, which is still quite concerning in a population study of quite a number of patients. That was echoed in other studies. You commented on the South American one. I wasn't there for that, so I don't know too much of the detail, but obviously Harry Rutter's presentation at the very start of the week on Wednesday evening really stressed the population style management programs and looking more at those levels than the individualized ones that we often talk about here with small studies. More and more data will be coming out as to population incidences of all sorts of liver disease, but particularly NAFLD and NASH.
And just to comment on that, the one thing that raises my ire more than anything else is the undercalling of the percentage of NASH patients, particularly in middle-aged patients that we see in clinical practice. I'm not talking about the 90-year-old nonagenarian that we might see, but we're not going to do a liver biopsy on them. I'm not talking about the 5-year-old running around, and I'm not saying they can't have NASH with bad disease. I'm talking about, as an adult physician, the people that I see in clinical practice, how common is this disease? And it's a whole lot more common than people realize. And I would just caution that unless you do prospective liver biopsy trials or some prospective measure that can be validated, doing these retrospective cross-sectional database looks at the prevalence and incidence of disease is going to undercall it by quite a bit. That's just my opinion, and I'm sure people disagree, but I just think we're minimizing the real prevalence of what's going on here. And I'll also say that we can make a big push at primary care to get patients to gastroenterologists, but having just come from two of my partners' offices across town today, until I have a therapy to put in their hands, they're going to say, go back to your primary care for lifestyle modification, and thank you for the upper and lower endoscopy that you provided to me by sending this patient forward. And what that's going to do is a negative feedback loop for my primary care provider is going to say, well, what good was it to send that patient up? Why should I even look for this until we have an effective therapy? So it's a vicious cycle.
Just to follow up on this point, and I think Stephen said it very well there, the danger with studies like that study coming out of Spain is that you never know the denominator. You acquire people with a liver stiffness of greater than 8 and you study some of those. You have no concept of what you've failed to pick up with an imperfect test, and therefore you will always underestimate it. That's one of the big challenges with that design of study, and the only way you can get around it is either to biopsy the whole lot, which is not realistic, sadly, or alternatively you have to follow them up and find out what happened. And those are the only two ways I can think of. I mean, Ian, Mazen, I don't know if there's another way you guys have thought of it, but that would be the challenge, in my opinion.
Yeah, so I mean, this is a very difficult nut to crack, isn't it? Otherwise, we'd all have solved it by now. And I think we're going to struggle to do any large-scale biopsy studies. So we've got to be settling large-scale prospective biomarker-based studies if, and I think it is if, we're going to do studies like this. Ultimately, it comes down to what we think liver disease that needs treating looks like and finding ways to best identify those patients, because in reality, we cannot be diagnosing 1 in 5 or 1 in 4 or perhaps even 1 in 3 adults with fatty liver disease and then going on on and risk stratifying them and telling the majority of them that actually this is a bit of the phrase from Southampton is this is a liver warning. I don't know what that means, and nor do I know how it's actionable to the patient. So we have to be clear what it is that we think is important, whether that's going to be patients that we're going to treat, whether that's going to be patients who we're going to follow up, all important questions. And this goes for, well, the two common liver diseases, so alcohol and NAFLD. And to bring us back to Louisa's point about pathways, one of the issues is that we've got two types of pathways at the moment, or three perhaps. One that looks at people who've got abnormal liver blood tests and aims to diagnose everybody. Ones that look at fibrosis generally in the population, considering both alcohol and non-alcoholic fatty liver disease. And a third that is risk stratification after diagnosis. And we need to decide, or ideally do studies, to figure out which is the best of those approaches in what context. Because at the moment we're providing mixed, sometimes complex messages to primary care, and I don't think it's particularly surprising that the uptake of those pathways is limited.
So going back to the point about you send the primary care patient to the hepatologist says, until you have drugs, I'm sending you back to primary care for diet and exercise. Will the best strategy change when we have drugs, when we have better drugs, when we have more drugs, and when we have greater faith in our non-invasive tests? In other words, is the right strategy for looking at patients, how strongly is it going to be correlated to what we can do for the patient and how easy it is to do it?
It certainly makes the argument for testing a lot easier if you've got a clear downstream strategy. Say, if I find this, then we're going to do, find X, we're going to do Y. The big question in the treatment piece that we didn't discuss is exactly what the indication for treatment will be and in what population. So the same time as the ILC was on, the American Diabetes Association meeting was on, and they've published a series of viewpoints in Lancet Diabetes and Endocrinology that accompanies their postgraduate course, and I'd encourage people to go and look at them because they are terrific, and they cover very diverse topics from how to intervene on highly processed foods all the way to complex management of type 2 diabetes. And there's an interesting discussion piece comparing the American and European Diabetes Association guidelines with the European Society of Cardiology guidance. And you can see there that the indications for the types of drugs that we would like to think of as commonly used therapies pending trials, namely semaglutide, SGLT2s, drugs that have a broadly pluripotent effect on the patient's phenotype, are going to be highly prescribed, and probably more so as time goes on. And at what point the indication for those drugs becomes a liver indication rather than a diabetes and cardiovascular risk reduction indication is unclear. And it seems to me that the greatest risk to the patient until they've got cirrhosis, sticking to the ordinal scales, but until they've got cirrhosis, is a cardiovascular risk indication. And so I think we've got to be able to articulate what is going to be important, the population that's going to be appropriate for a liver indicated treatment.
So since everybody's on mute at once, I'm going to take that as the last comment on that portion of the conversation, unless anybody has anything they want to add and we're serendipitously on mute or just being polite. With that, we're about an hour along, so I think it's not a bad time to go to final question and wrap up, right? And I think the final question is, given everything that we saw last week, and we can extend it back to the EASLD session before that, what message goes forward that actually changes the way we look at patients or look at disease now? And by we, I don't only mean the people on this call, but I mean more broadly the structure that engages liver patients and metabolic patients and fatty liver disease patients. What do you see as the most important thing likely to change in the short to medium run going forward? Nothing might be an acceptable answer, by the way, if that's the case. Quentin, go ahead.
I'll have a first go at this one. I think the most exciting part of the EASL meeting this year was the basic science seminar, the one day where we did a deep dive into multi-omics analysis, artificial intelligence and big data, as we start to leverage those to really go further into the pathophysiology that we're actually going to get some great treatments coming out. We're starting that journey. The trials that are already taking place are actually providing the data for that journey. But I think that's how we're going to crack this.
For me, and from a population perspective, I suppose it was Harry Ruttup. And whilst he was very eloquent in it, a lot of it was done and we've discussed before. But I think I get a sense of a general momentum. We've had a campaign in this country in the last 12 months and we've had a sugar tax, but we've had an obesity strategy. We've had a group of kids fighting the government to get advertising taken off mainstream TV for children, and I think awareness, and they called Bite Back 2030, and I think they have achieved something that I didn't think they'd achieved as quickly, and we now will be bringing in no advertising of junk food and sugar drinks before 9 o'clock in the evening to protect children. And I think this comes back to what Harry Rutter was saying about if you put obstacles in everybody's way, there's no wonder that we struggle to take the healthy path if you only give us the unhealthy path. So I think I'm generally feeling there is a momentum, particularly in this country, moving forward. I think it was covered in Easel quite nicely, and I'd like to see a momentum with primary care, with diabetes, endocrine, cardiology, where we increase awareness throughout and maybe COVID's triggered that as well, that people are more aware of their health, and it's not just what's on the outside that we can see, it's actually what's on the inside, which covers liver. So I'm generally getting a sense of a momentum, but it's whether we carry on that momentum with all of our research and all of our figures and the care that we can now offer these people.
I'm kind of glass half full. Despite all the problems we had, drug failing, biopsies problems, you name it, we're walking slowly but surely. Why I say that? We do have a drug that will likely achieve the primary endpoint in phase 3, which I think is going to be resmetirom. It does have likely good cardiovascular profile. That's going to be chased by 2 drugs that they have also good efficacy plus cardiovascular benefits. We also have non-invasive testings that they are being developed, moving the right directions. We have consortiums such as Litmus and Nimble confirming the efficacy of those. We have data now showing that they are correlating with primary outcomes and showing longitudinal outcomes. We have solutions coming for the biopsies while we are still relying on histology outcomes such as AI. So I think the future is bright. In 2024, 2025, we're going to start seeing solutions for patients. And it's just going to get better from there. So in the next few years, I think the field is going to start seeing solutions and we're going to build on them. So looking forward to that.
Yeah, I'll keep mine simple. I just think that we're advancing the field slowly but surely on disease awareness, on identifying the patients at risk for disease, coming up with non-invasive tests, addressing 3 different contexts of use, and therapeutics to manage those patients. To me, it's like going to the fair, each of us grabbing a water gun and squirting it into the race car to see who can get to the top the quickest. There are multiple different race cars all moving up that hill at the same time. And depending on which Congress we go to, one particular track advances quicker than the other track. But at the same token, they're all moving forward. And to Mazen's point, it's just a matter of time. We're not getting there as fast as any of us would have liked to be, but we're definitely getting there. And to Quentin's point, he's always the deep thinker in the room anyway, but using the omics platforms and really getting at understanding the pathogenesis of disease is only going to make the 2nd, 3rd, and 4th generation therapies that much better and provide a better opportunity for fixed dose combination therapy with minimal AEs.
I agree with what Mazen and Stephen are saying, really, and that is that the RLC, there was no big breakthrough. There was nothing, showstopper where you stopped and said, crikey, how have I missed not knowing about that for the last 3 months? So I didn't see it as being particularly exciting or thrilling. There was some good data presented, but it was building on existing knowledge, and particularly around the non-invasive testing. And I think that's probably where there was the greatest new data presented through synthesis of existing data sets and, new data generated in the consortia.
Okay, thanks. So my favorite moment of our 3 podcast, not necessarily the meeting, came courtesy of Louise. On Friday, we were talking about the Holmer paper, comparing intermittent fasting to a low-carb, high-fat diet. And at the end, Louise made the comment that I thought was really important and instructive, that there was a flaw in the study in that the level of support given the control group was so much less than the level of support given the trial groups, so that you couldn't really tell how much of that was about the diet and how much of that was the level of support. I mention that because after being stuck for decades, in a variety of commercialization models that were driven by the idea that the commercializers were selling food, the growth right now on the commercial side of this equation, outside of the meeting, is in people selling behavioral solutions to people who want to behave better. Now, I get to, people who listen to podcasts know my wife works for Noom, so I get to learn a little bit about Noom up close, but Noom is hardly alone, and more competitors are coming out, and other people are changing their models. So while we're looking to develop drugs that are going to be a little later than we want, I think the overall metabolic side and the side that's tied to weight loss, patients are getting better ways to deal with that when they want to. And if there's anything, and this goes back to the comment about why the control group was overlooked, if there's anything that we should be thinking about how to do more of and better of as part of this, it's to help patients who want to take those steps find their way to a behavioral platform that works for them. We don't have the drugs for them yet. We kind of have, we know some things about the diagnostics. It's not everywhere we want it to be on a drug level, but FIB4, as people have pointed out, is helpful in everyday therapy. And we have and ALT and a whole bunch of other things that we have readily available. So now the question is, while we're doing the heavy lifting that you folks described, can we at the same time do a better job of matching patients to behavioral tools that work for them? And I suspect that the way the world's going to work is that's going to be what fills in the gap until we get to probably resmetirom. I don't know how anybody else feels about that.
The diet will work very well with people like Stephen, who drinks Corona Light, versus other people that they drink the real Corona.
So, Mazen, since you've already told us that you want to be in better shape than you are, you better put down the Stella Artois.
Sorry, Stephen, I had to pick on you at the end.
It's sad that I'm becoming the old man of the
group. It has nothing to do with old. It has to do with the Corona Light.
yeah, dude, I'm older than you and I don't drink Corona Light. I'm way older than you and I don't drink Corona Light. At any rate, with that, let me thank all our panelists for, I think, a great conversation today. I'm going to cut this off right now, and then I'll come back and do the business portion when everyone's done. So thanks, Quentin, for coming back. Please don't make it more than another year before you get back here again. And everybody else, it's been great. Thanks.
Thanks, guys.
There's not a ton of business to report this week. Frankly, we've been too busy, too focused on International NASH Day and then Digital ILC 2021.
Live audience will become a permanent feature of Surfing NASH within the next month.
I'll get to our schedule in a minute. One thing we learned, though, is that live audience is a good idea for the folks who attend, and for us as well. We plan to introduce it as a weekly feature by the end of July. Keep an eye out for emails and posts on this.
Speaking of schedule, what's coming in July?
We're still working it out, but July should be fascinating. Over July 4th weekend, we will be recutting the ILC 2021 content, including the previews, same-day coverages, wrap-ups, and interviews, into topic-related follow-ups on 3 to 5 specific issues of interest. Depending upon how much content we create, these may actually replace the July 7th podcast, in addition to running over the weekend. One reason is that July 7th, I go on a 7-day electronics-free vacation, which means I will not have time to work on conversations for the weekend of July 10th and 11th if we drop a fresh episode on the 7th. Donna Cryer is guest hosting for me on the episode that records on July 12th and drops on the 14th. She will be asking for input from our audience on some of GLI's major upcoming policy and education initiatives, and we'll post more on this and send mail right after the July 4th holiday. Episodes on tap for July and August include one whose likely title is Why NASH Drugs Will Not Feel Like Statins to Payers, which named Alcori and I previewed during the ILC coverage. Another on the growing challenge of pediatric NAFLD, one on the new EASL guidelines released last week, and finally a celebration at the end of the month of our 150th episode and 1 year since our cutover to Buzzsprout.
How can listeners enroll to learn about live audience tickets when we make them available in the near future?
In our system, we email you an audience link, which is like a ticket, for whichever episode you wish to attend. We have 1,000 audience slots to hand out, so at least for now, don't worry about not getting a ticket. However, we need your name and email address to include you on our audience invitation list. So please go to the Surfing NASH webpage and click the link for audience requests if you want to make sure you're on our audience mailing list. And with that, I want to welcome Louise and Stephen back after they took some downtime during ILC 2021. Thank Mazen, Ian, and Quentin for a great conversation, and particularly to thank the magic man, Mike Wilson, for making the live ILC recording sound as good as they did, and as always to Eric and Polly for the things they do every week. Check for pictures on the website if you want to see Stephen's sand constructions during his time in Panama City. And with that, we will post the next episode on July 7th or 14th, depending upon how much material we find when we review all the ILC tapes. And until then, stay safe, surf on. Bye-bye now.
Thanks for listening. Have any questions for the surfers? Want to get your email address on the list for future live audiences? Go to the surfingnash.com website and click the appropriate link. Have any comment to share? Join the discussion on our LinkedIn and Facebook Surfing the NASH Tsunami discussion groups. See you next week on the Surfing the NASH Tsunami podcast.